Good evening again. That's hopefully better now. And I'm delighted to welcome those of you online and those of you in the room here as well to a talk by Professor Pedro Estrela, who is a professor of biosensors and bioelectronics. As a doctor, I've grown up in my career with increasingly sophisticated diagnostic techniques. When I first arrived in medicine, which was quite a long time ago, it was quite hard to get anything except the most basic blood tests, and even those took time. And then I think probably the first really revolutionary thing that arrived on the scene was pulse oximetry for measuring oxygen in the blood, which really did change the way that we could non-invasively manage our patients. But we've all had experience of rapid diagnostic testing. Some of that experience we might not have wanted to have in the last couple of years. But there is no doubt that having that has transformed the way that we have been able to manage the pandemic. And those kinds of technologies and other sensing technologies are going to revolutionize medicine in the future with increasingly sophisticated use and AI using biofeedback to manage illnesses in real time. And I'm really interested to hear what Pedro is going to tell us today about this growing area of technology, which is going to appear on our doorsteps very soon.
Right. Thank you very much, Mark. Thank you for the invitation to be here and talk about, I tried to give a nice title, medical analysis on the chip, revolution happening. I'm just trying to give a flavor of where we are and where we are heading. A lot of the things that I'll show are things that are actually already happening within some degree, but it might need some tweaking to actually get it into real practice. And also, then maybe also some ideas about why things are not necessarily being applied as much as they should. What I mean by medical analysis on the chip is basically having some sort of device that can measure as much as possible information about a patient or about an individual that to determine a series of medical conditions or just to monitor the overall health of an individual. For those of you who that are from my generation, you'll remember the Star Trek track order. And that's really actually this has inspired a lot of the researchers in biosensing field. Is trying to see, is there a way to actually get a device that can measure a whole set of things in one go in the real time? Don't think that we'll ever get the hands of Spock actually monitoring the patients, but the idea of having a device that actually measures many different things at the same time to actually determine the condition of a patient, that is something that is actually already happening. The whole idea of the Star Trek track order actually, as I said, has inspired many researchers. There has been actually a series of high-level competitions. There's one US track order challenge, which was at $10 million for the best products that could mimic what was doing. And the whole idea is to have this kind of real rapid analysis devices. I'm just showing a video, just basically illustrates briefly the type of things that we are looking at. This is actually research from a colleague of mine, and then animation from a student from Bad Spa University.
If the forward-looking arrow will be called investigated. of the anatomy. It scans the molecule, protein, DNA, the indications of disease. The blower extends from the gizmo towards the specimen to extract a sample of tissue. This sample is delivered back to the gizmo to be amplified. In the beginning, pass through the enlarged particles. The molecule monitor. The molecule monitor extrudes from the center of the gizmo. When a defective molecule is detected, a unique signal is displayed. The protein probe. The protein probe scans the sample for proteins and enlarges them for a clearer reading. The gauge highlights whether the protein is positive or negative on a defective scale. The DNA detector. The DNA detector picks up a sample from the specimen to survey the defective DNA. The sample is drawn through the examination tunnel, one base at a time, and its nucleotide structure is shown on the screen. When a defective nucleotide structure is identified, a unique signal is shown. The instant readout that PEG offers allows for rapid administration of treatment, and its portability and size makes it ideal for home diagnosis.
So this is just, of course, an animation, but is very much representing what we are doing in the field. We're talking mostly about diagnostics, so we're not talking about the whole system of management of disease. So we're not talking about delivering drugs automated way. The main reason for that is because there's some strong regulatory obstacles to do that. But we're mostly focusing my research and many others at the University of Bath, but around the world, is very much looking at how can we optimize these devices that we can measure a series of biomarkers from the body. And biomarkers are basically any proteins, any DNA, any small molecules that are present in the body that can be indicative of something happening in the body. And can be disease. A lot of the work we've been doing is mostly on early detection of cancer, but also looking at progression of cancer, and then a lot of work on infectious diseases and so on. And it's trying to understand how the body expresses these different molecules and how we can actually use that information to give real-time information back to the patient or to the clinician. At the core, so I'm going to talk about a series of devices. At the core of what I'm going to talk about is what we call biosensors. And I'll very briefly explain what biosensors are, but it's basically just any sensor that measures a biological entity or a sensor that incorporates some biological entity. So anything that to do with measuring proteins or DNA or glucose or lactate, etc. We get all based on biosensors. And then we'll talk about how this, using things like miniaturization techniques of both micro nano fabrication, but also miniaturization of electronics and use of microfluidics and use of then machine learning, artificial intelligence methods, we develop these full lab-on-chip approaches. And lab-on-chip approach is basically your track order, yeah, sample in and answer out. In a simple way. I'll also mention a relatively new field, which is called organ on a chip. I'll explain briefly what that is and what that can lead us to in the future. So all of this is basically to lead to approaches which are more personalized in terms of, I think, long are the days where the consensus that everyone with a specific type of disease is just given one drug, come back in 6 months and we see how it is. And I think there is more and more the need to personalize the treatment, personalize the approaches for each individual, so that there's either a stratification or there is actually close personalization of the treatment to the patient. Companion diagnostics is a big field where we start using these techniques for the patients themselves to monitor their condition. It's not necessarily to give diagnostics, but it's actually someone, for example, a cancer patient, monitor the progression of different biomarkers over time, so that they can get information at more data points, so they can do the test at home, just stores on a computer or goes to the cloud and goes to the server where the clinician has access to. It can look at the progression of the markers over time, so that it can then adjust the therapeutic interventions that they're doing. And then full medical management systems, where we can also start integrating the therapeutics with diagnostics on the same devices. So just very briefly, we're saying sensor. Basically, sensor is anything that measures something. That's all it is. So the idea is that any stimulus, which can be heat, light, sound, pressure, motion, etc., you then use some sort of mechanism to transmit that into something that we can measure. And the biosensor basically is just something that incorporates a biological entity. So when we talk about biosensor, we're talking not only about, let's say, what we call the transducer, which is what transfers one stimulus to an output, but actually is how we integrate that with a biological entity. For example, we'll know about the glucose sensors. And so a glucose sensor is an electrochemical sensor, which just has some enzyme that catalyzes glucose if it is present. That gives a rise of an electron, and then that gives an output, typically electrical signal. Of course, it doesn't have to be, can be optical or so. So when we talk about biosensor, we're talking about the whole thing. And we can have, sorry, you probably cannot see this very well. We can have a series of different types of, there's always a lot of different analytes that we want to measure. So it can be, as I mentioned, protein, DNA, but also if you want to measure bacteria, viruses, but also small molecules like lactate, glucose, etc. But also we use biosensors to measure other things. We use biosensors to measure toxins. We use biosensors to measure metals in water, for example, all sorts of water pollution. They use different types of biosensors for that. So we can actually measure many different things. And we use these bioreceptors, biocomponents, to measure that. Typically, we use enzymes and antibodies. So enzyme, like for the glucose sensing. A lot of the lab tests that you do when you take blood and they send it to the lab, a lot of those are antibody-based. In a sense, what we do is actually integrate those on the small device rather than having on the lab. And then we have these different types of transduction mechanisms, and applications are not only clinical, but also environmental, food quality control, and many others. Pharmaceutical drug discovery is also a big field. And what is becoming also very important is actual control of industrial processes, particularly bioprocesses. Now, typical examples, I already mentioned the glucose sensor. Is a very big, actually this dominates, or better, I'm actually not sure anymore. Pre-COVID, this dominated the market. Because glucose sensing, it's very important because there are millions of people with diabetes. They need to measure the glucose levels several times a day, and therefore there's been quite a lot of big effort to develop low-cost solutions for people to measure, to monitor this at home. There's also, of course, a commercial business aspect to it, that they might sell. They might be very low costs, but if they sell millions of them, they make millions of profits in a sense. Yeah. So it is a big market, and that's also what pushes the companies to move towards the glucose sensing. And pre-COVID, it was 70% of the market of biosensor was glucose, because it's a money-making machine. There's another classic example, which is a pregnancy test. And that is a very simple device where you just basically use capillary flow approaches to have the fluid, in this case urine, to go through a system that then there is accumulation of basically is an antibody base, and there's accumulation of HCG on specific areas, and that gives a change of color. And I'll show you briefly how that works. And of course, like the glucose, like the pregnancy tests, COVID lateral flow tests is actually the same principle. Is actually very old technology, just applied with a different for a different application. This, of course, you all know, and we all use regularly, or used to use regularly. Well, I still do, but even though I know that they actually don't work that well. Let me rephrase this. If they give positive results, it's positive for sure, almost for sure. But if it is negative, it doesn't mean much, because there's a lot of issues with sampling and so on, which is not controlled. But these type of devices are very low cost, and the reason why they're very low cost is because of what we call passive devices. You don't need to have any power to drive any of the, because there's no electronics in there. Now, and there's been also a lot of movement towards looking at continuous monitoring. So these are the sensors that are measured, something that you measure a single point, but continuous measurement, particularly for glucose, is extremely important, because if you just measure several times a day, you actually might miss peaks of hyperglycemia or hypoglycemia, which can be quite important to understand if they are present or not, to control the condition of the patient. So continuous monitoring is becoming very important. There was some early attempts to do some small subcutaneous sensors. There was a GlucoWatch, which this kind of collapsed, but now it's coming back with a new version of it. And many of you might know of the Abbott Freestyle Libre, which basically just has a device that just stays there for 4 weeks, 7 weeks, 7 weeks, I think now. And you can just approach a device or a mobile phone to get the readout every time you want. Now, a lot of the approaches that we use on developing this type of sensors is basically looking at what's happening in nature and try to improve it. Classical example, what we call electronic noses. You will see often scientists give fancy names for things that are actually quite simple. It's just a gas sensor, but it's fancier to call it electronic nose. It's thinking about what happens in the body. So you have a sample that goes through the nose. You have the mucus and the cilia, and then you have which are basically the receptors of different molecules that are in the sample. And then that signal is transmitted through some cells, through the olfactory nerve, and then there is a kind of a firing of different neurons that goes into olfactory cortex. And the olfactory cortex basically just looks at all these things that were fired and tries to combine them and get some sort of a recognition of what happened there. I mean, it's like the combination, basically olfactory cortex has a memory bank of a combination of different molecules that associates to different things. So if you smell pizza margherita, it's not like you have a receptor in your nose for pizza margherita. You just recognize the different molecules that are present there, and you have your memory to say, okay, the combination of these levels is that. And that's basically what we try to do on the chip. We have an array of sensors, each one picking up a different molecule. And then we have a preprocessor that basically looks at which of these sensors were fired up or at which level they were. And then we have some pattern recognition, looking at data that's been added to the system to recognize. And then you start seeing, okay, that one corresponds to this type of for example, bacterial contamination. We don't actually detect. If you smell milk, you see that it is rotten on the sensor. You actually don't detect exactly that. What you detect is like things like volatile organic compounds that are expressed there, or if there's contamination of food, you detect specific bacteria that are there. So, but gives you at the end the same information. And then we use also quite a lot of nanotechnologies or nanomaterials to improve our devices and our sensors. And the reason why we do that is mostly, it's not because it's fancy, also because it's fancy, but not only because it's fancy, is that actually there's very important properties. And most important properties of nanomaterials is that they have a similar size to the molecules that we actually want to detect. So if you look at things like the DNA and proteins, we are talking on the nanometer scale. Okay, if you go to viruses, you go to 100 nano, bacteria, micron, and cells, a few micron. And if you look at the different types of materials like nanotubes, nanowires, nanoparticles, we are basically on the nano to micro scale. Why is that important? Well, it's a question of scale. If I put my hat on this table, the table is not going to fill the hat. I mean, if you look at the overall mass of the table, it doesn't change. If I put my hat on a small piece of paper, then there is a big change on the overall mass. And that's basically what we measure. We measure changes. So the fact that we are doing something which is of the similar size of what we want to detect actually makes the signal very, very strong. Also, nanomaterials, it's mostly surface and little volume. That means that we can just play what's on the surface to change anything and measure it because it changes the overall properties of the material. And nanomaterials have been used actually for a long, long time on biosensors. Actually, the pregnancy test uses gold nanoparticles. The COVID test uses gold nanoparticles. And I'm just going to give you a very brief video showing how a lateral flow device works, because since it's the first of these lab-on-chip approaches. I apologize for the music, it's not my choice. But the idea is that in this case, it's for using blood samples, so you can put some filters there to filter out the red blood cells, so that most of the molecules go through into the nitrocellulose, basically this paper that is underneath. And because its paper is going to very quickly wet, in a sense, yeah, we add some buffer that is just to help the flow and also to minimize the influence of these red blood cells. And then what you have is an area where you have some nanoparticles, gold nanoparticles, with an antibody. And as the flow goes through the paper, it just pushes these antibodies to go with it. If your sample has the analyte that you are looking for, it's going to bind to the antibodies, and you have other antibodies that is going to bind to something that you have already prepared on a specific area, which is going to be your test and control lines that you'll know for the COVID tests. As the particles accumulate, so if the test is there, it's just going to accumulate. If the analyte is not there, it's going to just flow through and go to the test line, which is further. When the particles get together, because they are gold nanoparticles, if they are just dispersed, basically you see a very light yellow color. When they accumulate, you get very close together, they get red. It's a property of the nanoparticles. So what you see, and that's the reason why actually you see red stripes on the COVID and on pregnancy tests, is because it uses gold nanoparticles. When they get very close together, the color changes. It's an optical property of the nanoparticles. And this is exactly the same thing that is used for the pregnancy test, but there we use urine and antibodies for HCG. In the case of COVID, you basically use, you have a swab which you then put on a buffer, and then just goes through and just measures protein on the virus. Now, what I'm going to talk about mostly, I'm going to just give a few examples of the type of sensors that we do here, or in my group here in Bath, and they are mostly electrochemical sensors. I apologize for those that work more on the photonics aspect of it, but there's many different types of sensors that we can use. Electrochemical sensors have an advantage, which is a sensor or a biosensor that basically gives some sort of electrical signal. And partly because I'm in the electrical engineering department, but partly because it's actually something that the technology exists to measure variations in any electrical property is actually easy to integrate into real devices. And that is, behind it is just the concept of electrochemical double layer. I'll not go into the details, but the main idea is that if you have a metal electrode in contact with a solution which has some ions on it, like blood or urine or saliva, etc., which has ions, then at the interface between the metal and the solution, there's the formation of what we call the double layer. It's basically just accumulation of charges on the electrode and then counter ions at a certain distance. And that creates a dynamic system which basically has a separation of charge, which from the electronics point of view, that just means a capacitor. If we have some molecules there, like an antibody, then there is something that goes and binds there that is going to change either the charge of your biomolecule, or it's just changing the thickness, which has different properties than the water or than the blood or urine where you're measuring. And that can give rise in changes in either in potential or resistance or capacitance, or like glucose sensing, for example, it's an enzymatic process which just releases an electron in the process. So you have a change in current. So these are all things that we can easily measure, and we use different techniques to measure this. So kind of electrochemical sensors, which is quite relatively straightforward from the electronics point of view. So basically, what I want to say is that almost any interaction between two molecules gives rise to a change in one of these parameters that we can measure very easily. We also use, I'll skip this very quickly because it's actually not that important, but just to mention that although we use antibodies for many of our sensors, we tend to try to improve on antibodies. And the reason why I say improve antibodies is that antibodies are great, but they are not so great for a sensor. The reason for that is that when the body produces antibody against something, it produces an antibody against a class of molecules. It's not one for every single small variation that you have, for example, just to give an example, prostate-specific antigen, which is a prostate cancer-related marker, but it's actually a prostate marker more than a prostate cancer marker. And actually, there are small variations on the structure of this protein that are quite indicative of, or can give information about if high levels of this molecule are indeed because of cancer or not, or what is the stage of the cancer, if it is indolent or aggressive. In the case, for example, of prostate cancer, it's very important to understand that, because most of us will develop prostate cancer, hopefully at a very late age, and the elder you are, there's the more question mark of should you treat it or not, because the treatment can be worse than being with the cancer, especially if it is indolent and not aggressive. So these molecules, they have different small variations, either on the structure, they might have some extra sugars, or they might have some small variations in some what we call epitopes of the protein. When we produce antibodies, the antibody does not distinguish between them. So there is a series of synthetic molecules approaches to actually get molecules that do discriminate between these small types. And one of them is DNA aptamers. There's also peptide aptamers. There's also molecular imprinted polymers. There's quite a few approaches that we use to actually do that, to recognize in a more specific way. Also, antibodies, from the electrochemical point of view, antibodies are too big. And when we say too big, it's 10 nanometers. We already think that's too big. These ones are tend to be more than a few nanometers. So that means that whatever happens in terms of binding events happens very close to our electrode, and that gives us more information. And also other types of synthetic molecules that we use. I'll not go through that. Just to give a few very quick examples of the type of work that we do here, just to show that we can measure different types of biomarkers in body samples in very easy way. Well, not very easy. This takes years and years to develop, of course, but once it's done, it's done. For example, this is just using actually an approach that uses gold nanoparticles just to improve what we call surface density. And we then basically have some of these molecules, synthetic molecules that just captures PSA which is present in the solution. And then we can look at these variations in resistance of the system. And we can measure with these sensors down to picograms per mil. That doesn't tell you much, but the clinical range is 1,000 to 10,000 times that. So we are much below what is needed in a sense for clinical range. Actually, it's too good of a sensor almost. But we can do that, and we can do that for many different molecules. Also, an example, for example, I was mentioning the PSA that has some sugars on the surface, which actually gives information or can be used as a more clear biomarker for prostate cancer. An antibody does not distinguish if it has this sugar or not, but these synthetic molecules, we can make them to distinguish. And we can look at, then measure at the same time, the total amount of PSA that we have in the blood and the amount of PSA that has these sugars, and then look at the ratio. And I'll go this very quickly, but this is basically showing using clinical samples from healthy individuals, with just benign tumors, and with cancer, prostate cancer. I don't know which stage this was, but we can see that by measuring both of these things at the same time, actually we can get a very good correlation, distinguish those that have aggressive cancer and those that have indolent cancer. Okay, it's not perfect. There's a small point there that it's just annoying us, but that's life. But this is just to show that this type of approach is very easy. Now, what is of course, sorry, just give you another example. Besides proteins, there's also something called microRNAs, and microRNAs is just basically small pieces of DNA, of RNA, that are expressed in the blood and in what we call exosomes, because many of them are also in the blood, and that's the levels of these molecules, they go up and down according to the stage of cancer. So actually it can be used to distinguish at which stage the cancer is, and also can give us an idea of what is the likely progression of the cancer. We can use this type of techniques also to look at detection of these molecules. We go down to very low limits of detection. We can go measure very well down to what we call femtomolar level, which is basically what we need to measure in clinical samples. But of course, clinical samples, blood samples are very complex, and we have not just one type of microRNA, we have hundreds or thousands of types of microRNA, so sometimes you get interference. So we can then start using other approaches. For example, we can use magnetic nanobeads. So it's basically some nanoparticles which have magnetic properties, and we can modify them and throw to the sample, to the serum or the blood sample, and just mix that, and this basically going to fish and attach only the ones that we want to get. And then we just use a magnet to pull them out, and we concentrate everything. So what we get at that stage is just these magnetic nanobeads with the sample that we want, and everything else stays behind it, and we just discard of. And then we can just either try to measure directly or just remove it and put it on a sensor and detect the levels that we have there. And this we can do on what I'm going to explain in a bit, you can do on kind of a lab-on-chip approach where you can start using this type of mixers and sample preparation on the same device so that we have just sample in, answer out at the end. And then just, I think this is probably the last example, just using other type of micro electrodes, some microfabrication techniques to get some very high area between the electrodes. Basically, this is what we call finger electrodes, integrated electrodes, where we just basically have two electrodes, but they are kind of like fingers, and they just go like that. So we have actually a lot of area between the two electrodes at the microscope. And if anything happens on the electrodes, then it changes the capacitance of the system between the two electrodes. And we can use that, for example, in this case, we put a molecule to detect HER2, which is a breast cancer biomarker, and we can get very nice changes in capacitance of this molecule in blood serum with a very high, what we call dynamic range. That's basically well within the clinical range, what is needed for clinical applications. Another example, just because I've been talking about biomarkers, most that we can detect, but actually we can use the same techniques to measure other things. And one work that we were looking, initially we were interested in cancer therapeutic drugs to see how does the patient assimilate the drugs. So that if you can measure not only the markers, but actually also the levels of the drug in the body, it can also tell if the dosage is correct or not, or give some indication of dosage and so on. We could not get our hands easily on an anti-cancer drug, but we went for tenofovir, which is a antiretroviral drug used for HIV, but not only, for many other type of infection. And yeah, don't worry about that scheme. That's just because it's from a different system. But we can detect this in serum. Again, having this drug in serum, we can detect the concentration of this in serum. And this is quite important because we can start looking at, we can measure the proteins, we can measure the microRNAs, we can measure, I did not show here, but we can measure what we call circulating tumor cells, we can measure many different things, and we can also measure the drugs that are being used by the patients. And that is quite important because it's a lot of information that is there. And if you just monitor the progression of this, actually gives a very valuable information to the clinician about how the therapy is going, whether or not should change the dosage, whether or not should change the drug, whether or not should go for some another totally different type of therapy. Instead of using these drugs, you're going to radiotherapy or going to whatever it is required. So it's a lot of information that we can actually get. The last example I have here is actually measuring uropathogenic E. coli. So a lot of urinary tract infections are caused by this bacteria, and it's often quite when people go to hospital with a urinary tract infection, that's the first thing that tests is if it is E. coli or not, because the type of antibiotic that is given then depends if it is bacterial infection or not. The problem is that there are many different types of E. coli. Some of them completely harmless, others are cause this urinary tract infections, and it's important to distinguish between them. And we used a similar type of electrochemical sensors to actually detect the presence of this E. coli, uropathogenic E. coli, and we could measure down to relatively, well, I'll say good levels. I'll not say very good levels, but we could distinguish between pathogenic ones and non-pathogenic ones. And what this means is that with this type of devices, this one might still need maybe 1 hour of culturing of the sample to get enough bacteria to actually measure. But we can improve that, and other people have improved that, so that they actually can get a test that is immediate. At the moment, it's taken to the lab, and normally it's the next day you get the result of whether or not it is uropathogenic E. coli. And that is problematic because what it is done in most practices is that you actually start the patient on some antibiotic, some wide spectrum antibiotic, which might not be the most adequate. And also creates, I don't know if you have heard of, it's a very, very big issue at the moment, it's antimicrobial resistance. And one of the big problems is that you get more and more resistance and more you use wide spectrum antibiotics, and then they stop working. And if they stop working, then you need to go to other types of antibiotics, and there's not that many actually out there, even after all this century of development of drug industry. So understanding immediately what type of bacteria it is very important to get the right treatment and avoid problems in the longer term. So in my group, we've been doing quite a lot of different types of sensors, as I mentioned, proteins, microRNAs, circulating DNA, circulating cells, also different markers for infectious diseases, and all of these electrochemical sensors, we can basically integrate them and measure many different things at the same time. It's just an electrical signal. I mean, we can, if you look at this screen here, you have, I don't know, 100 million pixels. I don't know how many pixels it has, but basically you switch between them in milliseconds, yeah? So you can switch between 10, 20, 30, 100 sensors in milliseconds, well, less than millisecond, in microseconds. It's very easy. From electronics point of view, it's very, very easy to do. These are all just measuring electrical signals. So as long as you have each sensor measuring one different thing, then you can just switch, read them all very quickly. And so you can start putting them together, and that means that you can also miniaturize, and then having the readouts that you can measure a panel of these markers at the same time. For example, for cancer, it is very important to measure a panel of markers than a single marker. There's not a single marker that is good enough for any type of cancer. You need to measure a panel of them. So this is quite good for diagnostic disease, for prognosis, to try to understand that, or understand how the disease is likely to progress. People that are under surveillance because they have some predisposition for some type of cancer, or family history in some type of cancer, or something that needs to be checked over time because a mold that is appearing and is changing quickly, or whatever. And also for monitoring of treatment, for example, in patients. And then we can use this type of synthetic receptors to enhance the sensors to make more specific and get to better diagnostic and certification. So we've done quite a lot of work on many different types of sensors, yeah, don't worry about that. But yeah, we've been working mostly on cancer diagnostic, cancer management systems, but also HIV management systems. That is actually a quite interesting project that appeared. And I should mention only because one of the things that more and more we do when we develop sensors is actually having what we call co-design approaches and participatory research. It's trying to understand actually what the people need, the end users, what they actually need and what they will likely to use. So HIV tests, they have been around for a long time. They're actually relatively low cost. But there's a big problem. I was with some colleagues in South Africa, working with some NGOs in South Africa, and we went to visit different places in South Africa to see how they actually do HIV testing and how they actually monitor the whole process. And we were very, very surprised to see that, well, they have the most amazing screening program in the world. It's none better. They basically screen almost the entire population, which is really remarkable. But there's a problem. We were, for example, in some areas, which is the Transkei region, which is an area which is very beautiful but very, very poor. And it's you basically have huts where people live. They have one here, another one is 2 kilometers away, and the other one is 3 kilometers away. So it's very, very dispersed. And the nearest clinic might be 20 kilometers walking from that. So they might go and get tests. And if it tests positive, then they need to take a blood sample. And the blood sample is sent to a lab to check the levels of CD4 plus cells, and also to check kidney function. because a lot of the antiretroviral drugs, they actually affect the kidneys, and because there's a lot of problems of malnutrition and so on, they need to check the kidney function to see that it's not going to harm, and if it is, then change the drug. And that takes 1 day. They are not coming back. 60% of population, they don't come back in these regions. So that means that 60% of the population that is actually infected with HIV continues to transmit infection on their local communities. So there's a big, big problem that. So this is a project we are doing with collaboration with in South Africa, is actually get some tests that if you test positive for HIV, then do another quick test to just measure kidney function and measure the levels of CD4. So that you can then start treatment immediately. But we've also done some work on detection of malaria, to test tuberculosis in sputum, Chagas disease, urinary tract infection, and so on, different type of things. It might look a lot, but actually the techniques are more or less the same, and we can just then adapt. It makes it sound easy, but it's 20 years of work. But as I mentioned, they are all techniques that you can miniaturize and multiplex. Multiplex means just measuring several of them at the same time. And we can create some chips, but basically we can measure everything. Now, when we go to try to use this, of course, the more sensors you have, then the more complex it becomes, of course. And you also have to think, okay, we have a sample that we want to take. We don't want to bleed the patient to death. We want to just take a small droplet of blood, like a finger prick, and go into measure many different things at the same time. So how do we do that? Well, we create this, what we call lab-on-chip approaches. And the first part of lab-on-chip approach is the use of microfluidics. And microfluidics has one very, very big advantage, is that, and there's a kind of a short animation video. I think we still have time to show it. But it's basically, it speeds up things. It's not only using lower volumes, but speeds up things because everything is closer to each other. So the reactions are much quicker. And that's basically what this video tries to show.
Ready when you are. When I started out as a common figure, the research game was pretty one-dimensional. But wow, how that ever changed with microfluidics. Microfluidics, the manipulation of small volumes of liquid. It brings the game down to a smaller, more precise and more intense scale. It is creating a real buzz and is becoming an important field of scientific research with intense action taking place in the microbioreactor. We are here at a training complex where microfluidic coaches put new tactics into action to stay on top of this scaled-down game. Since the rescaling of stadiums with reduced space on the field, teams play in closer proximity. This means more tackles and action at a lightning-fast pace. Did coaches have to deal with this in the slow old macro stadium days? No. Does it present new challenges? You bet. Take new conditions like laminar flow, a core principle of microfluidics that forces the players to play a more organized and precise game. This coach has players moving in perfect parallel formation, a slick display of undisrupted laminar flow. Ready for the tackling now. And look at that. Absolute control of collisions. Mixing and the speed it takes place is an essential part of the game. And this coach has it sorted. Oh, super. Let's go back and see more advantages of the new game. Resources were expensive with the macro. Now it's cheaper and there's less wastage. Before, it was one game at a time. Now with parallelization, there's a feast of action with many games going on simultaneously. Experimental macro play was slow. Results took ages. The speedy microfluidics game brings faster and more reliable results. Microsystems are much safer and better organized. It's a breeze for coaches to control whole teams at once, and the smaller scale provides a perfect training ground for smooth heat transfers between players. In the macro days, similar tackles could easily escalate into dangerous chaotic situations. This results in less chance of injury for those involved. New tools like computer-simulated games help coaches predict and adjust to scenarios that might occur in a microbioreactor. But we all know that microfluidics is more than just a game. It's the products that come out of the microbioreactors that are what's really important. Countless applications beneficial to humankind. New medicines and devices that improve the lives of people with medical conditions. Or solutions to solve our pressing environmental problems such as greener energy and cleaner drinking water. Microfluidics even helps us saving our little mates, since it reduces animal testing. This is the future, and that's why I am not only a commentator, I am a fan.
So this is just to illustrate the power of the magic of microfluidics. You see, indeed, you can get things done much quicker because the reactions happen much quicker. You don't have diffusion processes. You don't have the issues of gravity. You have things happening very, very quickly, and the molecules interact with each other in a very, very quick way and in a very controlled way. And you can use minimal volumes to actually do that. And that is the beauty of using that. So what we do on the lab-on-chip approach is basically integrate the sensors with microfluidics, with electronics, and we can do a lot of things with these devices. We can have different types of controls to make sample processing, to make chemical reactions, and have, of course, also the sensors and so on. A colleague of mine is actually developing a kind of lab-on-chip approach for COVID testing, amplification COVID testing. At the moment, you take a sample. Okay, yeah, you can get a PCR test in some places down to 1 hour, 2 hours if you pay a fortune for it. This device at the moment is measuring in less than 10 minutes. Amplification, same level of amplification, and same level of accuracy. And this is because we do that at a micro scale, and things are always just much faster. So the amplification process is much faster. And there's quite a lot of systems out there that we can people have used. That you can have already even the needle for the finger prick. You can even integrate on the device so that the blood goes directly into the system and measures different things. You can have electronics there. We can have antennas and so on, so you can actually transmit the data also to a mobile phone, for example, so that you can store all the data and so on. So that is things that happen. So this is actually an old device from iStat, which is then bought by Abbott. And here, basically what they have is just a cartridge, and then we put this drop of blood, and that goes to a handheld device. This is the approach that we're actually using. Instead of integrating a lot of the electronics and the displays and the communication and so on the chip itself, we do the lab-on-chip only to the point where we put the sample and you have the reactions, and then you insert on a handheld device so that all the electronics, everything stays there. The reason for that is that if we use blood, you cannot reuse your chip. So to minimize cost and minimize wastage, we tend to use this approach so that the chip itself has the minimum function and just integrates with a handheld device. Measures, take it out, throw it away, in a sustainable way, of course, and then use the next one, and so on. So, and that's basically what happens with the glucose sensors. Yeah, you have this strip where you put the blood, it goes to a handheld device, then take out the strip, throw it away. And the handheld device just basically stays there, reusable all the time. And so this is basically what we are all doing. It is this type of approach. And we can, as I mentioned, have all these different techniques, electrochemical techniques, but can be other types of techniques that can have on this type of devices. And we can have our cartridge, we have all different types of sensors measuring using different molecules, synthetic or non-synthetic. And then you basically get a readout of different markers at the same time, which can give information. I mean, this slide was done for a cancer type of approach, but it can basically depends what you want to measure. You can measure it. And just very briefly, I want to mention another area where it is working quite a lot. Many people working quite a lot, which is so-called organ on chip. We are not harvesting organs on a chip, but almost. In the sense that organ chips, basically what you do is that you take cells from a patient, for example, it can be from a biopsy or whatever, from a specific organ, and you culture that on a chip, and you control the conditions of the cells and control the environment to mimic what happens on the organ. And then you can just start seeing the influence, for example, of the different drugs on the cells of the patient. There's a series of drugs that you might know that work for that specific type of disease, but you might not necessarily know if they are all going to work or not. So you can create these systems where you basically create the cells and you create different chambers. So it's a kind of 3D structure. We have different chambers basically mimicking what's happening on the organ. You can have also channels which mimic the flow, the capillary flow of blood underneath, for example, a series of cells with some membrane and so on, and just keep on monitoring what's happening. And this is basically what is being used, or pharmaceuticals are very much interested in being used for what we call personalized medicine approaches, where basically you are targeting the therapeutics for that specific patient. So you use the cells of that patient, you put them on a chip, you culture them, throw different drugs, you see how the drugs affect, you see how the cells respond to the different drugs, and then you know that, or at least you have an estimation of what sort of drug will work for that patient. And the next patient might be slightly different and different concentrations. So you actually have a very personalized approach for this. And this is really becoming very, very big. And there are some groups doing, trying to do the body on chip, basically having different chambers with different cells from different organs so that you can basically do those. It's like, I'm almost finishing. Just to mention, I will not spend time on this, but just mention that actually a lot of these approaches we use also for other, not only for medical, but other applications, particularly in terms of anything that can help to improve public health. Water is the main one. Water quality monitoring is extremely important. Well, that is done in the Western world by the water companies before the water goes into your tap. But in many places, actually, that control does not exist. So there's actually a lot of devices being developed, lab and chip devices being developed that can monitor the water at the tap, for example. And even the water companies here are interested in that type of approach because they will have an extra level of control and satisfy the regulations also, of course. Another big field is so-called wastewater-based epidemiology, and that is basically looking at what is on wastewater. And the reason for that, I'm going to skip some of these slides. The reason for that is that basically wastewater is a pool of samples from the local population. It has other things, but it's a pool of urine, a pool of feces from a local population. And urine is full of biomarkers. And it's not only health markers, actually it's all lifestyle markers. It's from drugs, from pharmaceutical active compounds. Most, when you take some paracetamol or some ibuprofen, most of it actually is not assimilated by the body, it goes through the urine. And that is also a big problem for the wastewater treatment plants because it's not so easy to get rid of those, and you don't want them back on the water system. Yeah? So we can use this to actually look at what's happening at the local community level, use this type of approaches to measure what is in the wastewater, and that gives information on what's happening at the local community, and that can be for many different things. We've done some. So there's a big group in Bath working on this using some special techniques, mass spectrometry techniques, and so on to measure whole range of different compounds. We developed some sensors to measure just a few because it's too big of a challenge in a sense. But we've done some quick work just measuring some markers and some drugs, and also the people are working quite a lot on COVID detection in wastewater to pinpoint if there's any outbreaks of COVID in specific communities. Just to show you one example, we did a small device that was measuring cocaine in wastewater, and this was samples taken from the same place. I'm not going to mention where it is. It's not Bath. It's nearby, but not Bath. Samples taken every day of the week, and we ask relatively high levels of cocaine every day of the week, and then we see the recreational use over the weekend of cocaine. And so we can use this type of information to give information about what's happening at the local community level. Drugs is one thing, but of course there's many others, and as mentioned, COVID is a bit of work. So just to finalize, we can. I didn't show a lot of examples of lab on chip, but basically I show you the bits that make the lab on chip approaches, and we can think about personalized infection management systems, for example, where you have samples taken from a patient, and then that goes into a lab on chip, which basically does some processing, there's some sensing, and that gives you a kind of a fingerprint of that patient. Before that, for example, what we can do, if we take, let's say, COVID, we see a lot of samples from many patients, and we know how the disease progressed. So we can actually get that information and create and feed into a machine learning artificial intelligence algorithm that actually tells you what is likely to be the profile of that patient, for example, in terms of immune response, to see if that patient is likely to is likely to be severely infected or not by COVID. And there's a lot of information out there. So we actually just need to feed that into a kind of a algorithm, a machine learning process to get some classifiers so that we can then have few sensors, measures, sample in, measures, and then use these classifiers to actually say, yeah, this patient is likely to get into hospital very quickly. This patient is unlikely to get severe disease and so on. And then having kind of a feedback loop with the clinician that make the intervention. As I mentioned, companion diagnostics is very important, particularly cancer diagnostics, cancer management, where people can start sampling at home. We're all happy to do COVID tests at home, so why not do this? And I think that there is more people willing to actually do this type of interventions. So anyone that is under surveillance or in treatment can just do these tests now and then at home. So there's finger prick, put a sample in, and you get information about different biomarkers. And you do that one day, and you do that any X time. Number of days, and that basically just logs information. It's actually not diagnosing anything. It's just giving information that can help then the physician to decide on the treatment. And so the future is basically that type of approaches, and start integrating that to actually with other things, wearable technologies. A lot of people are wearing Fitbits and so on, and actually keep on monitoring different things. And we can start integrating all that and actually get a lot of information. The reason why I put a question mark is because there are some challenges there. And the main challenge is that with wearable technologies, we can measure very nice things, but there are some things we cannot measure unless we go down into the skin and go to the blood side. And that is one of the main problems. People talk about wearable technology solving everything. Yes and no. But still, I believe that there's a lot of work, for example, being done with the microneedles. And we saw, for example, the Abbott Libre pad for glucose, basically just as a needle that goes in interstitial fluid and actually goes to the first small blood vessels so that I can get the accurate result. But so there's many technologies out there that people are starting to use to extract samples through the skin in a painless way. And that can be done in a wearable way. And then you can basically have a whole loop where you basically gather all this information and it can trigger immediately, because you can have the machine learning, artificial intelligence algorithms to trigger alarms for different people in terms of can be either to the family or call the emergency room or just give information to technician. So the future, whether or not it is a lab on a smartphone, I don't know. We'll see. Might be, but we'll see how it goes. just few issues to consider. As I mentioned, sampling is important. When I mean sampling, I mean where do you take the sample from? That is very important because for example, for many cancers, we could try to go to measuring sweat, but actually that doesn't give you information. Sweat is good for measuring things like glucose, lactate, and so on, but for proteins is not good because most proteins will not go through the skin. So we might need to go to interstitial fluids. For some might be okay, others might be okay. There's still not enough information in a sense about that. So we might need to start looking at different measures. So depending on what you want to measure, you might need to sample different parts of the body. And that is a problem, is try to understand how that goes. And then the integration of this, all this type of biosensor, but with other physiological and physical sensors. That is the future, to actually start looking at the holistic approach of the patient, or of the person, to get all the information that can give extra information and extra prediction of what might happen, might not happen. As I mentioned, for many diseases, we cannot look at single biomarkers, but we need a panel of them, and that means that we need to have different correlation established. For example, on cancer stages, how the correlation between different markers is achieved. And this is a problem because we don't have enough information from stage 0 patients. That is those that might start having some sort of lesion, but actually don't have cancer yet. And we don't have enough data for those. We don't have data when people are actually diagnosed. So we need to start getting a lot of these also called longitudinal approach studies to actually monitor people over time, even the healthy people over time, to get that enough information that can tell us, okay, yeah, now I can see that this starts appearing at a specific time of the cancer progression, and that can give you an early indication of what's happening. Dementia, for example, is a very, very interesting case where there's a series of markers that seem to be expressed at least in CSF, but also in blood. And so you can actually start getting some information, but we don't have that much information about patients that do not have dementia and that are on onset of dementia, to actually know exactly how much of these molecules are actually present in blood at a very early stage. But people talk about being able to detect dementia maybe up to 20 years before people develop dementia. Whether or not you need that information, that's a different question, and I'll leave that to the experts. But there is a lot of information, even before we have any sort of symptoms, that we can use these type of techniques to look at. We have a lot of data that can be collected and sent to the cloud, and so there's a lot of data analytics. That also poses a question is, which data to give to the clinicians? And when we talk about clinicians, we need to think about, are they in primary care, secondary care, or tertiary care? Because actually they want different sort of information. And having need for different sort of information might need that we need different sensors or lab-on-chip devices for the different applications. And also, very importantly, what information should we give to patients? We definitely don't want to give a device that they measure at home saying you have cancer. Cannot have that. So what is the minimum amount of information that gives confidence to the patients to actually do the tests, but without actually causing anxiety and so on? So other issues to consider is acceptability of new technologies. That's why we use quite a lot of these so-called co-design approaches, participatory design, so on. Basically, we engage with the people that are going to use the sensors at the very beginning to understand what they are willing to use, and also to give us advice of what they want and what they are likely to use. There are some examples. There was some old study that someone was developing a rapid test for tuberculosis. And they went to some small village in Tanzania and so on to do a test that, because they had some connections through that. And they went to the local community, and just everyone refused to do the test. And that was for a very simple reason, just that the box of the test was red. Red was symbolized of demons. And therefore, they were not willing to do that because that was a demon type of thing. And even these small things can completely destroy. And if you don't have a proper dialogue with the end users, you might end up, and I'm not going to go into that discussion, but you might end up with a GM discussion, which never really happened. There was just a complete condemnation of GM, of genetic modified foods, without actually fully understanding the benefits and the cons. I'm not going to say that I'm pro or con, but what I'm saying is that if you don't have this dialogue at the very beginning, you might just be creating technologies that will never be used. Of course, there's problems with data security and data privacy. You're generating a lot of data that's telling a lot about yourself. Are you willing to share that? Are you confident that it is secure enough? There's a lot of ethical issues with all of this, particularly as we start going towards the companion diagnostics to be used at home, and of course, regulatory aspects. So, diagnostics on the chip is actually already here. In different scales and different applications. It can go much, much further, but we need to basically have a kind of a looking at the whole problems to solve in order to actually get there in a meaningful way that is actually meaningful for the end user, being the patient or being the clinician or whoever it is. So it's quite try to understand who is going to use, why they are going to use it, and is there a need to use it? Also, that's also an important question. So I just finish with the question.
So thank you very much. We'll take questions from the room, and if people want to put questions online in the chat, then I can read them out. And any questions in the room, I'll kind of paraphrase into the microphone so that people online can hear. So, any questions? Anna?
I have a question. When you started talking about Africa blood, I'm sure you've heard of the Theranos case in the United States. So I'm wondering, I mean, if anyone doesn't know, I'm sure you can explain it better than I can. I'm wondering, you and your colleagues, when it came to light, obviously that was a massive fraud, but the thing that I know about it was that Silicon Valley got incredibly excited about it, investing humongous amounts of money. I think it was the next thing. But were you and your colleagues, if you followed it from the beginning or around that time as everything was coming out, were you skeptical that it was even possible what they were hoping to do, or were you hopeful? Was there signs from in the academic community that that kind of thing was not possible at the moment from the beginning, and it was smoke and mirrors?
Okay, that's a very interesting question. So Theranos is a very interesting example. So Theranos basically worked on the premise that you have someone promising using the magic of microfluidics to detect everything and convince the investors to give a lot of money. The scientific community, we know it is possible to do that, but no one ever saw any data coming from that lab. So we were all very skeptical. Not that it cannot be done, but that they have done it. And the investors actually never went to the academics to ask, what do you think? So they just, because also there was a forging of a series of letters to give some sort of credibility to what they were doing, that convinced the investors. But that's one of the problems, that often investors, they get just like the words like magic. But yeah, that's the only scientific claim that I ever heard from her is using the magic of microfluidics. Yeah, that doesn't say anything. For the scientific community, yes, we were concerned that there was not enough there, but we are all confident that this can be done. So whether or not they have done it, we did not.
Any other questions?
Sorry, just a small parenthesis. That Theranos actually made a very negative impact to the field of diagnostics and this type of diagnostics because people just started to mistrust everything. In that sense, COVID was I don't want to say a good thing, but COVID gave a push to say, actually, yeah, the premise of looking at things and start measuring things at home and with a small drop of blood and so on, or saliva or swab, whatever, actually works, and people are willing to do it. So that changed out, because it did affect quite a lot, particularly in terms of investment. And unfortunately, I have to say, it was particularly companies led by women that were mostly affected, which is shocking, but that's the situation.
That is in the meeting. Right. That's it.
It's already out, maybe. I did.
It's in.
All right. Oh, yeah. I knew they were doing a video, but a movie, but I didn't know.
Basically, it's live footage. You go through it. It's really fascinating. I think right. Set on the movies.
Any other questions?
With the example of the kind of the whole body on a chip, so regarding the kind of actual feasibility of, you mentioned before you could take a select sample from an organ, but how do you do that if you're creating the entire body? Do you work from like stem cells or?
No, you need to actually extract cells from it. So it is a bit problematic. So the full body on a chip, it's only maybe useful for example, cancers of unknown origin, for example, or there is a condition that simply cannot really pinpoint where it comes from. So only then it becomes really useful, that type of technique. You'll try to get cells from the organs, but that means biopsies, which for some organs might be quite tricky. But yes, if you don't have that, then you do the next best possible thing, which will be stem cells and so on.
Sample view.
Yes, absolutely. That's what you start with. I mean, that example there with the 10 organs, I don't think it's ever going to be used. It's just a show off of technology to say that it can be done.
Not all on a chip could be going from getting a sample to getting a useful result out. Are you talking days or weeks of preparation to start testing?
For the organ on chip, it might be a question of days if you kind of know what you're looking for, you kind of have an idea of what sort of, for example, if you are all you want to determine is what drug to give that patient or dose to give that patient, then yeah, it's a question of days. If you actually want to do something more complex to actually understand what's happening more at a molecular level or cellular level and so on, that is kind of longer term studies. Also, there's people looking at very long term studies, basically to see also after, for example, the trigger of the mechanism that cancer cells communicate, to see if actually they become dormant or actually are killed. If they become dormant, what triggers when they come back to life and have recurrence of cancer. So you can just start then looking to keep them on an incubator, keep on measuring, feeding them. But yeah, I'll say might be a question of days. But for many conditions, that's not a big issue. If you talk about cancer, if you're talking about infectious disease, that's a different thing. You need something much faster.
Are you getting enough funding? Because before COVID, well, I was absolutely shocked. I've worked in diagnostics for years, that the government said we don't have a UK diagnostics industry. We've got to go abroad to get it fixed. And now, post-COVID, I see everything going into cell and gene therapy. They're so boasting about that all the time. So I'm hoping that some funding is, and especially, unfortunately, from the EU and cut off from Horizon programs. So what's the situation now?
So questions for people who didn't hear online is questions about the level of funding for this kind of technology and research.
Well, I should say that the diagnostics industry in the UK is actually quite strong at the academic and SME level. It's just that the government does not necessarily see that because their friends didn't know the right people. I think that they could have invested much more on actually what was happening. And there was investment, actually, I have to say. There was a lot of investment during COVID. There was a lot of investment onto academic research, but also on industrial research. There was a series of programs from NHK to SMEs and so on to develop. I actually have a startup with a colleague which got some of that funding, and then they are now not involved so much anymore, but they are now developing kind of like the lateral flow devices between electronic readout, and they start developing that to other applications, like pregnancy tests for blind people and so on, that kind of thing. So there was funding there. There was just everyone. The problem is that they cut the funding for everything else, which means that we all had to change our what we were doing into COVID, which, yeah, maybe it was not the right thing. Now there was then two problems: is the as you mentioned, the lack of access to European funds. That's a very big problem. And there's another problem which actually stopped before that, which is when the government stopped the Global Challenges Research Fund. And because that was funding, that was, I should not say this online, but basically we're double counting the money that they're using for international aid they use also for research. But that actually enabled us to tackle things with countries like the work I've been doing with tuberculosis and malaria, Chagas disease, and so on. That was all done through that, in a sense, through these interactions with countries in South America, in Africa, in India, etc. So that actually helps quite a lot in terms of the development. But that stopped. There's no more international aid of that sort. Well, there's almost no international aid, but anyway. So at the moment it is challenging, but the funding is kind of slowly going back into what it was before. Whether or not that's good or bad, I don't know. But it is maybe more focusing on big things rather than small things. And that is a bit of a problem, I think. But I'll say that funding does exist. And particularly if you have an entrepreneurial spirit, then you can at least start interacting with the right people to try to get that to the next level. And then there is quite a lot of funding, I would say, for SMEs and so on to take it to at least prototype stage. And then sell to the Americans. Yeah.
So I have a question, and there's one online as well. My question is, I suppose I recognize three broad categories of testing. The first is the patient who has symptoms where you're trying to make a diagnosis. The second is disease monitoring, the patient already has a diagnosis. And the third is the pre-symptomatic identification of disease. And you alluded to that as sort of from an ethical point of view. But I wonder from a scientific point of view whether there are opportunities using these sorts of technologies, perhaps combined with AI, to identify these markers early on.
Absolutely. But the problem with AI is, so AI is great, but AI, people say AI will solve everything, but AI will solve everything if you have good enough data for it to solve everything. Because you need to train, you need to give enough data. And that is what I was saying, that we do need a series of longitudinal studies to actually understand from the very early stages, give enough data to actually tell the machine to say, actually, this is a signature that something is going to happen in the future. And I think that is the main problem, is the lack of data for that. Because why should we? Why should I be testing for prostate cancer? Well, I should, because I'm over 50. But why should I? When I'm 30, I'll say, why should I? But maybe there's some indication already there.
Testing for diseases, like most dementias where there is limited treatment, you hit an ethical issue at the same time.
Exactly. Because dementia is a very interesting case from the ethical point of view. It's very, very interesting because what does it? Unless you give enough tools, or you have or develop enough tools to at least slow down the progression. But that is a good thing. But is it worth monitoring that 20 years before you have symptoms? Maybe not. Because you're just putting the patient with some mental health issues, basically.
One another question.
You can get some of that information anonymously. So, for instance, like institutes like Biobank, which stores samples and then links that to future medical diseases. So, have you used that as a resource to find people who have developed diseases 10 years later, and then go back to look at those samples? Have you used those? Sort of
Yes. So people do use biobanks all the time. But even on biobanks, it's very limited storage of samples from healthy patients, and patients that actually people that go over time, because you need the sample before they had a disease, and then when they started getting symptoms, and then how they progress. So you need all these data points from the same person, and it is difficult to get that. So you can get a lot of samples and anonymized samples and so on with some information that can help. And we use that. We do use that, or many people use that for all sorts of diseases, but it's limited amount of information also.
So we've got a couple of online questions now. First one is from Tom Eldridge. Great lecture, thank you. You mentioned nanoparticles are already used in these sensors, gold nanoparticles in particular. How relevant are carbon nanotubes and graphene in this field?
Very relevant. Well, no, I'm laughing because we discussed before the lecture. Was asking about the same thing. Extremely relevant, depending on what you want to do with it, of course. So there's a lot of you can improve the sensitivity of many of these sensors, for example, using nanostructured materials like people have used with carbon nanotubes, and particularly with graphene. If you need to improve it, but often you actually don't need it. For many of the examples I showed, we are well within the clinical range with the sensors we used, using just standard electrodes, which means maybe there is no need to go down the route of nanomaterials, which might be more expensive in terms of mass scale production. But for some applications, absolutely, they are wonderful. For others, maybe it's an overkill. So it's a question of thinking what is the market and what you want to achieve with it. But yes, absolutely.
So we have Andy Morley. He's raised his hand. Well, this will be complicated to get him to say what he wants to say.
Hello. Can you hear me? Okay.
We can hear you.
Oh, great. Hello there, Pedro. This is Andy Morley. I'm actually calling from Germany at the moment. We've spoken in the past, I think back in 2015, with regard to diagnostics for cancer, and we talked about PSA and all sorts of different things. But one of the big challenge is really at the end of the day to do anything very exciting was resources. And one of the things I'd like to ask you with regard to that and sort of being able to move forward in this sort of complex environment in terms of getting resources. We have some exciting activity in terms of the NHS and these integrated care systems. Now, one of the interesting aspects of these new models of care is that they are supposedly going to have diagnostic hubs, which are going to start to pull together some of the things that you've covered today. And I wanted to know if you were aware of these particular diagnostic hubs, as I think this is potentially an opportunity to do some sort of public health sort of diagnostic work that would be very valuable along the lines of companion diagnostics and monitoring, and also pulling in data. So that was one question. The other was, I'm about to have a conversation, or in fact tomorrow, with the Institute of Cancer Research in terms of data science and looking at how we can pull things together. I wondered, I think there's a significant opportunity in terms of joining the dots and coming out of the silos to see whether we can exploit data and the science that you've just sort of elaborated, so that we can actually move forward in a really quite good, exciting way.
Yes, thank you very much. The Diagnostic Hubs, that's new initiative which is quite welcoming. I am aware of it. I'm not involved with it at the moment, for many reasons which I don't want to go into details here at the moment. But it is, let's just say that they have some preferential partners, and I'm not one of them yet. But I am aware of it, and it's something that it's on my agenda to start to try to get more involved with it. And I think it is a great thing. I think there is a kind of still a bit of a limitation from the NHS in terms of what they actually want to see there. The NHS is very slow at adopting things for many reasons. I'm not criticizing them necessarily. They have the reasons why they are slow. And I think even on this initiative, which is very, very welcome, I think that the range of things they actually want to do there is somewhat limited. And it will be good to talk with them to see how they can expand that at least in a cost-efficient way, which is, at the end of the day, that matters very much for NHS, also for obvious reasons. Data, yes, it's very important. It's something that it's more and more, as I think if we talk in 2015, maybe I was not so much worried about data back then. I'm very much worried about data now. So I think it's maybe it will be good to actually have a chat again to discuss that. So I guess you have my email, just drop me an email and we'll have a chat about that. I think that there is quite a lot of opportunities to start bringing new techniques. The NHS is very much in favor of artificial intelligence. They believe that's going to solve all their problems. Which is a good thing, which means that they have the right attitude towards it. Maybe they hope too much from it, but we can use a lot of these techniques, data analytics techniques, into starting developing complex sensors that don't actually measure what we were looking for, in a sense. What showed that idea of looking at the barcode or fingerprint of a patient is very much about not necessarily looking at specific disease, but looking at how the patient, for example, immune response is like, how the patient's proteomics or metabolics is to actually understand, are they predisposed for certain disease? Are they likely to have certain disease or respond better to certain type of treatment and so on? And that involves a lot of data analysis. We can only measure so many things. We need to actually have the data analytics to get meaningful information from all the few things that we can measure. And so I think there's a lot of things that can be done and should be done and will be done. If we just have a chat. So I'm looking at the screen. And if you drop me an email and we can have another chat, we'll be very interested.
Thank you. So I think you can see from the level and nature of the questions, you've inspired a lot of interest, a very exciting field that I think is going to change that generation's healthcare. Perhaps not so much for me, but maybe a little bit.
Even yours.
Thank you very much. It's been a very, very interesting evening.